All right, welcome everyone in the session on non-biological complex drugs. It's a great pleasure for me to host you today. And of course, I speak on behalf of my co-chair, Scott McNeil, and all speakers today that we have loved to see you in Basel this year again. But during the circumstances, I think we're all glad that we can meet in a digital way. My name is Vlieger. I'm coordinating the NBCD working group, and as every CLINAM session and the conference, we're happy to host our science-based discussions within this conference. So, what are NBCDs? A quick reminder for everyone who must have seen this earlier in earlier sessions. A non-biological complex drugs is a medicinal product whose physical chemical properties cannot be fully characterized or quantified. These properties can also be substantially altered by the manufacturing process, and together, consequently, impact product performance. On the right-hand side of this slide, you see a number of examples, and this directly shows you why the CLINAM conference on nanomedicines is so relevant for this type of products. So today, it's either breakfast time at some of you, lunchtime or dinner time, so we prepared a 7-course menu for this afternoon. We have a group of speakers really going from the basics towards the regulations, science, and different complex products. And we end up as a proper CLINAM session with questions and debate. So some notes of administrative characteristics. We have 10 minutes of presentations by each of the presenters. And I will keep you to that and warn you when you still have 2 minutes to go. We do all questions at 6 p.m., so at the end of all presentations. Please submit your questions anytime during the session following the Ask Question button on the lower end of your screen. My co-chair, Scott McNeil, will guide you through the discussion and question sessions at the end of this presentation sessions. So a quick reminder for all speakers, remember to mute and unmute yourself. Share your video and your screen as we just practiced. I'll keep you to the 10 minutes time limit. And of course, in the end, at the end of the session, Scott McNeil, my co-chair, will read the questions to the speakers. So with that, I'd like to take no more time of this precious session and introduce our first speaker. That is Professor Dan Crumlin. Dan is a professor emeritus from the Department of Pharmaceutics of Utrecht University. He has a long-standing career in liposome formulation. He's co-founder of Octoplus. He has served on many scientific advisory boards, and he's still active as an advisor to industry, SMEs, and functions as an expert witness on the topic of complex formulations. Dan, please, the floor is yours.
Thanks, John. Thanks for giving me the opportunity to talk. Sort of an interesting title, Where to Go with Nano? Question. You see in the bottom these 2 gentlemen, well known for those who are have reached a certain age, arguing all the time, and that is exactly what happens and what leads to this title, Where to Go with Nano. Because there's something playing in nanoland. The scientists are in hot debate. Questions are being asked. Eyebrows are raised. Public debates that are set up. Institute funding is affected. Critical conclusions are drawn. In other words, there's a lot of discussion there. And discussion is sort of around these things like the success of nanomedicine. There's no question mark there. This is from Twan. But there is a question, exclamation mark here, cancer nanomedicines, so many papers and so few drugs. Translating nanomedicines, thinking beyond materials, that's the youngsters among us, thinking about what is nanomedicine really bringing to us, and what has the nanomedicine sort of wave or hype, as some people call it, what has it brought to the patient in the end. We wanted to do some fact-finding there, and we is Peter van Hogevest, Gert Storm, and myself, all the 3 are active within CLINAM, and we wrote this commentary for journal controlled release, The Role of Liposomes in Clinical Nanomedicine Development, what now what. You can find everything that I'm going to talk about in this brief presentation. You can find that in this commentary with a lot of more backup facts and data. But from the abstract of this commentary, I took these sentences. Research priorities for the future will be outlined based on experience with the most successful nanomedicine family within the broad field of nanomedicine so far, liposomes. An analysis of currently clinical tested, approved, and marketed liposome drug combinations provides these insights. Well, there are a number of points here. First of all, liposomes, the claim is the most successful family. And the other thing is that what is the contribution of nanomedicines to this development of this family. Well, let's first have a look at nanomedicine. When was the term coined and presented in literature for the first time? And that is when you go to Scopus, and when you look at this nanomedicine in these sources, then you'll find that around 2000 the nanomedicine term was used for the first time in literature. So that's only 20 years ago. And now when you look at liposomes and run the same trick, you find that in 2000, there were already 30,000 documents on liposomes around. So that's when nanomedicine started. So liposomes were far earlier than the nanomedicine era. Here we find again the presentation the picture that I showed you before. Conclusion, the liposome science was already well developed at the time that the term nanomedicine was coined. Well, the science might have been there, were the products already there? And then we have to go to, before I go to that, we have to go back a little, because I wanted to clarify the point that the liposomes are the most important and most successful family in nanomedicine land. And of course, what I say is not important. It's important what the FDA says. And this is a publication from FDA authors, Distribution of Nanomaterial Use in Drug Products from 1973 to 2015, number of submissions to the FDA. We'll find liposomes as the leader in the field, 1/3. This is good old nanocrystals, so oral delivery systems. Then we have emulsions, iron complex drugs. But liposomes are definitely in that era the leading nanomedicine, although nanomedicine, the term was not there yet. I know you can say, well, that was the past, but when you go to the nanomedicine days, and this is the period 2010 to 2015 that they analyzed, and you see that nothing has changed. It's still 1/3 of the application submissions to the FDA that were liposomes. So here my point, liposomes are the most successful family. Yes, in terms of submissions to the FDA, if you count it like that, that's true. The other thing is the science was developed before 2000. So now the next question in Dan, what about the products? What about the products? Were they then developed in the nanomedicine era? And now things become a little bit more complicated. Here you see liposomal formulations approved in the EU and US, a selection of the most successful products. There are two old ones, two before 2000, and that is the Embosome and the Doxil Celix. They were developed and approved before 2000, so they were before the nanomedicine era already on the market. The ones that are framed in red, they are liposome products that were marketed from 2000 on. The pain relief, anti-tumor, antimicrobial. And then we have an interesting one that is the Shingrix, that is liposomes used as adjuvant, so not so much as drug carrier, but as adjuvant. And I put patisiran there, which is admittedly not a liposome, but a lipid siRNA product, but I put it there to be complete. So indeed, in the period 2000-2020, you see that quite a number of these liposomes came to the market. And not only the innovative products, but also two generic versions of doxorubicin by Dr. Reddy's and Sun Pharma. A point that I would like to point out here is that when you look at the money side, here it's clear that the two old ones, they bring in as a revenue about $1 billion a year. None of the others come at that level. Maybe because they were approved later, maybe they are not so successful, but the two old ones are still the leading in terms of revenue. There's one exception there, and that's Shingrix, because Shingrix, with the adjuvant, that brings in about 800 million euros. So that is a real success, but you can argue what is the role of the liposome there. So point here is that, yes, there were products before the nanomedicine era started. The old ones are still the most successful ones. And the point I want to make is that when you look at all these different new ones, that the basic technologies and the analytics were developed well before 2000. So the technology was there, and that means that we now know that the nanomedicine field maybe helped in the development of these new ones, but the technology was already there. Well, is that true for only the liposomes? When you look at the other nanomedicine families, then you see that the FDA list provided by the Mello also points out nanocrystals, iron polymer complexes, drug protein complexes. It's all the same. The basic products, the major products, and the technologies were already developed in before 2000. Well, having said that, what is the future? We were talking about liposomes in the future, and now what? And here it's interesting when you go to the clinicaltrial.gov and you look at the pipeline, then take phase 3 clinical trials. Liposomes are by far the leading family there. And nanoparticles are hardly any in the phase 3. Abraxane are not there. But here again, when you look at what's in the pipeline and what nanomedicine will be leading in the future, then it's clear that liposomes will play a role there that cannot be pushed away. Then finally, something about what is Big Pharma doing here. When you look at the clinicaltrial.com data, then there's some involvement on Big Pharma. We look at the clinical development of these new medicines, some, but very little. Big Pharma is involved, but the major goal there is that they integrate, try to integrate the approved nanomedicines like doxorubicin, liposomes, etc., in existing treatment programs. So that is where Big Pharma plays a role, not so much in new ones, but in working on existing treatment protocols. Exceptions, GSK with the adjuvants, and of course, Enzyme, Moderna, we will talk about that more, we will hear about that more, and BioNTech. So in the mid-sized pharma companies that are dealing with nucleotide delivery, yes, they will definitely work on these nanomedicines on the basis of lipid-based technologies. And with that, I would like to finish and and go back to John for continuation of this discussion.
Wonderful. Thank you very much, Dan, for a great introductory talk and to show us what's actually happening and what we can expect. I'm sure you're pushing questions through the webpage. We'll collect them for the later part of the meeting. Dan, if you can stop sharing your screen, we can ask the presentation from our next speaker online by Susanne Bremer-Hofmann. Susanne is representing the European Commission Joint Research Center, and she's leading the lab responsible for nanomaterials. She's very active in the field of nanomedicines, for example, in the REFINE project, which I'm sure she's going to present some data and ask us some questions. Susanne is a very well-recognized expert in the field and always very open for discussion. Susanne, please, the floor is yours.
Thank you, John, for your nice introduction and giving me the opportunity to share our newest or progress of our research. Next slide, please. Okay, as you also already outlined, I will talk about mainly about REFINE. That is the Horizon 2020 project, the European project, which has started in December 2017 and will end next year, maybe in the beginning of 2022. The aim of REFINE is to improve the risk-benefit assessment of medicinal product and medical devices based on nanobiomaterials. And here we are looking at different aspects. So we are interested in the information requirements. So what is needed for the regulatory community to decide about safety and quality of a material. We are interested in methods, how we can address these information requirements. And actually in REFINE, we have also experimental work ongoing. Then we look at standardization requirements. So what kind of what kind of standardization needs are there for or have to be fulfilled for the various methods. We bridge communities, so we are in contact with the regulatory community, but also with the academics and industrial partners. And then the harmonization needs, because it's very important that we understand the international dimension, because we want to bring the products on markets of the various regions. A highlight of the project is the decision support system. That is an automatic tool which will help us to develop integrated testing strategies. Next slide, please. So one major outcome of the REFINE project is the so-called white paper. We have identified a number of challenges through surveys, questionnaires. We have discussed with the regulatory community. We have looked into the manuscripts and publication of regulatory scientists, and we grouped 6 different challenges. So we have, for example, the selection of the regulatory path that is in particular interesting here in Europe, because this depending on the mode of action, the product will be follow the regulatory path of medical devices or medicinal products. We have also here, that is Europe specific, we have the EU definition for nano-enabled medical devices. So this requires additional methods and approaches. Then we have the assessment on follow-on products, and I'm sure we will hear about that. And then we have the harmonization of regulatory practices. I mentioned that already. But now I would like to focus on the regulatory information needs. So what is needed to make sure that the product is safe, and also how we can address these regulatory information needs with suitable methods. Next slide, please. So my colleague, Blanca Halamoda, she has done a tremendous work and looked into all kinds of guidances that are available for medical devices and medicinal products from Europe, but also from the US and from Japan, and has extracted the information needs. And then we characterize these information needs in physical chemical parameters, so what is specific for nanomaterials and should be assessed, and also for the biological characterization. So here we come up with the bioburden. So there are some specificities for sterility and endotoxin levels. We have the pharmacokinetic parameters and also pharmacodynamics. Next slide, please. But we were asking ourselves, is there not more in in the literature may be hidden with regard to safety and quality. So can the scientific literature serve as an additional source of information? And we heard already from Dan that the number of publications with the term nanomedicine is increasing tremendously. So we have more than 1,000, even 1,500 publications just with the term nanomedicine each year. So we started, and this is not possible to go through manually. So we thought maybe automatic text mining tool could help us with that. And we started a little feasibility study. We used web-based search engines and searched for specific toxicities of the nanomaterial and have extracted more than 1,000 publications. Then we did different steps of curation, segmentation, and we also looked into the quality of the publications that we have extracted, and these and we use quality criteria that have been previously published by the Mirable Consortium. And at the end, we had around 500 publications in which we now look a bit deeper what kind of information we can automatically extract. And how that will look like, I show you in the next slide. So the information that we get, this is just an example, is here, for example, the various toxicities and how they are occurring with, are paired with the different platforms of nanomedicine. So we see that metal-based nanoparticles are triggering more toxicities than, for example, liposomal products. We were interested in immune effects, so we looked a bit deeper into the immunological aspects. Next slide, please. And here we could then identify various toxicities specific for pointing to immunological effects. We had spleen toxicity, accumulation in spleen, macrophage activation, hypersensitivity, inflammasome, and so on. But this is just an example. Once the database has been set up and the tools are available, you can also ask which kind of methods are used. We have compared in vitro, in vivo, for example. So these automatic tools allow a lot of possibilities to keep overview what's going on in the scientific literature. Next slide, please. Next slide, please. Of course, now we ask, do we have methods to address these identified information needs? And again, my colleague Blanca Alamora, she was looking into all kind of different standards that are available from ISO, from CEN, from ASTM, and came up with these kind of graph. This has been published last year. And what we see that the level of standards is rather low. So we looked into standards of medical application, but also for industrial application.
2 minutes, Jean.
Yeah, looking at this, we have seen that we have 5 different gaps of methods that have to be standardized in the field of kinetic properties, so stability, the ATME, the drug loading and drug release, for example, the surface properties, and also immunological responses. Next slide, please. So we wanted to know, again, are there methods out there that we can identify, prioritize, and suggest for further standardization, but also to understand what kind of methods should be developed. And here we quickly realized that we have different kind of gaps. So for example, we have methods available that are suggested by the scientific community, but we don't know whether they are relevant for the regulatory community. Then we have gaps where we do not have methods at all. So here we certainly need method and method development programs. Then we have methods that are available but not standardized. So here I'm thinking on the NCI, NCL, and UNCL methods. We have then methods where they are material specific, so we need to understand, can they be used for other materials? Then we have methods available that are not for nano. And then we have also standardized methods, but from other sectors. So again, we are doing, next slide, please. We are doing a big overview that is research in progress, again, in the immuno field, where we are collecting all this information and try to identify methods and also their validation status. Okay, next slide, please.
I have to ask you to round up, Susan.
Yeah, but I think the next slide is rounding up. Okay, so I highlighted that we have major challenges. I showed you the potential of text mining tools and highlighted the lack of standardized methods and that we need to bundle understand our standardization efforts. But I would like to close with reaching out to other sectors and would like to show that we have the Refine is organizing next week the Knowledge Exchange Conference, in which we are sharing knowledge and practices and efforts. Okay, so last slide, I think it's highlighting our feedback, and I would really like to get feedback from on our work from the various stakeholders that would help us to set our priorities and also push the regulatory science of nanomedicine forward. Thank you.
Thank you very much, Susanna, for an excellent overview. And also, it's great to see that the European Commission is so committed on this topic. And I'd really urge everyone to look at the Refine website. There's an open consultation currently ongoing, and indeed provide feedback to the Refine team on the questions Susanna just raised. So again, questions for Susanna, please write them in the section below. We will address them at the end of all presentations. Thank you, Susanna. So our next speaker is Mark van Eldijk. He is group leader and project manager at Ardena API and Nanomedicine Development and Manufacturing. He's actually based in Austin, Netherlands, in the former Chem Connection site. And before joining Ardena, Mark was a postdoc fellow at California Institute of Technology, and he has a PhD in biochemistry from Radboud University in Nijmegen. Mark, please, the floor is yours.
Thank you for the introduction, and thank you for the opportunity to share some of the experiences we have in overcoming upscaling of challenges for nanomedicine. So before I go into that, I would like to briefly introduce Ardena, because I could imagine not everyone is aware of us. So we are a contract development and manufacturing organization based in Europe. We guide our clients through the drug development and drug approval process, and we do this both on drug substance and drug products. So we have teams of experts working on small molecule API development, manufacturing, and on drug product, often solid dosage forms like capsules and tablets. But we also have a team of about 50 experts working on nanomedicine development and manufacturing, and that, of course, is the topic for today's presentation. Besides this chemical and pharmaceutical support, we also offer bioanalysis and dossier development support for the nanomedicine projects that are running at Ardena. So for the nanomedicine, we define them as medical application of nanoparticles. And we actually distinguish two routes of preparation. One is the bottom up, and the other is the top down route. So for today, I'll just be focusing on the bottom up application, where we build the nanoparticles from the individual constituents to build into the particles. So that's applied, for instance, for lipid or polymeric or metal oxide nanoparticles. So generally, the projects that are running at Ardena are for particles that have a diameter of smaller than 200 nanometers. and these particles are often used as drug products, as a drug delivery device, or a medical device, which could be imaging or tissue engineering. And as mentioned, we manufacture them in a bottom-up approach, and then we often make use of these principles mentioned here on the slide of self-assembly or a precipitation approach for manufacturing. We also make use of, for instance, crystallization, where we could make metal oxide nanoparticles. So the nanoparticles we produce can range from relatively simple to fairly complex. And here I'm showing one example of a particle where you could see many layers, so core, shell, encapsulation of API and equipment with targeting handles. So that's a fairly complex nanoparticle platform. And the experience we have with the types of nanoparticles is quite broad. So we have experience with solid nanoparticles like metal oxides, gold, silica nanoparticles, but also the more traditional nanoparticles like liposomes, but also polymeric-based and protein-based nanoparticles. And of course, we do this for our clients. And we have now already manufactured more than 20 different GMP nanomedicine products. And I'm here just listing a couple of clients that we are collaborating with. And some of these clients you might also see at the CLINAM summit. They are presenting their work here also. So very interesting to be collaborating with them. So our approach towards nanomedicine development is that we generally start with a technology transfer where we take the technology from our clients and implement that at our location. And often that's a small scale technology. So then we get to the next stage of development, where we perform both process development to scale up, often from milliliter scale all the way to liter scale and beyond 50 liters sometimes. And we also perform analytical method development and eventually validation of the analytical methods. After the completion of the development phase, we can go into manufacturing of tox batches and GMP batches. So, of course, the title for today was challenges in development. So some challenges that we experience are, for instance, in the scale up of certain batch-wise processes, for instance, for self-assembly of polymeric nanoparticles. Nanoprecipitation could be used to prepare those, or lipid-based. Liposomes can be made using lipid film hydration, but these methods are not very scalable. So for that reason, we designed a flow manufacturing system, which is very versatile and allows us to, in an easy and flexible way, scale up for manufacturing of these kind of nanoparticles, so polymeric and lipid-based. So this system works in a way where we dissolve the polymers or the lipids in a water-miscible organic solvent and mix that in flow with an aqueous solution. Then we have the option of downsizing through extrusion, for instance, for liposomes, or in-flow sonication could also be used in combination with polymeric nanoparticles to obtain the nanoparticles. One of the issues here still is, of course, that we have presence of organic solvent, and that's another challenge in upscaling. So in that case, so at small scale, you'd be able to remove the organic solvent through a process of centrifugation, dialysis, or lyophilization, but that's not feasible at larger scale due to cost or scaling limitations. And for that reason, we often go to tangential flow filtration, where we can purify nanoparticles up to 100 liter scale. And just very briefly, it's the passing of a nanoparticle solution over a membrane, circulating that, and then due to an overpressure, you're able to remove the solution, so the solvent, through the membrane here, and at the same time, you're adding in new washing solution. So basically doing an active dialysis, which is a really nice way for scaling up of purification of nanoparticles. And we apply that in pretty much, I would say, every project that we are running. Other challenges are in the analytical development, like also Suzanne was already mentioning in the previous talk. So you need to define the critical quality attributes, so the attributes, the list of attributes that potentially impact the quality, safety, or efficacy of the final product. And that's often divided in chemical composition, impurities, size distribution, and solution and microbiological characteristics. All of these methods, of course, also need to be validated. And if we look at a liposome example where we, for instance, make a liposome out of 2 lipids and a API lipid in flow manufacture, then perform tangential flow filtration workup, eventually filter the product to obtain a low bioburden bulk drug product solution of liposomes, then these would be the typical specifications. And here you have your chemical composition. So that's quantified for lipids by UPLC. So we quantify each of the components. We look at the impurities related to the components, but also related to the process. So related to the ethanol that's used, residual ethanol, and the elemental impurities, but also size and size distribution we can analyze using DLS or AF4 moles, and solution and microbial properties are also analyzed according to standard pharmacopoeia methods.
2 minutes, Mark.
Yeah, thank you. So these liposomes are a fairly easy example because they can be analyzed through dissolution of the individual components, as it's a non-covalently linked nanoparticle, and that's also true for polymeric micelles, for instance, if not cross-linked. But this, with this increasing complexity, also it becomes more complex to characterize the particles thoroughly. And that means you often need a more complex sample preparation. So this is also a challenge in the development of nanomedicine. So after we've overcome these challenges in the process and analytical development, we are able to manufacture a non-GMP batch that can be used for preclinical evaluation in GLP tox studies, after which we can also produce in a similar manner the GMP batch according to GMP compliant procedures. And then that batch can be released for use in phase 1, 2, or 3 clinical studies. So just to wrap up for today, I've really only shown the capability that we have for nanomedicine, so nanoparticle manufacturing and formulation, but we also have capabilities of synthesizing the precursors, coordinating fill and finish, labeling, packaging, bioanalysis, all of that for nanomedicine. But I hope I've given you some kind of flavor of the challenges that we typically experience in the nanoparticle manufacturing and formulation part of this trajectory. So thank you, and thank you for your attention.
Thank you very much, Mark, for an excellent talk, and also for staying within the time frame that we have provided you to present this work. I think it's really, really good to see that there's so much experience in the upscaling of these complex type of projects. And I think we all realize that there's a long way to go from the first, let's say, lab bench batch towards having enough material in great quality for clinical work. And you've shown a large number of different types of projects that you have experience in. And it's great to see that some of these actually are in clinic, as we have seen in the CLINAM conferences. So thanks so much. Any question to Mark, please write them in the section below the screen, and we'll come back to that at the end of the session. It's my pleasure now to introduce our next speaker. It's Donald Parsons. Donald is a Vice President, Early Technology Development and Lipid Nanoparticle Process Development at Moderna Therapeutics in Massachusetts, the US. Don is responsible for the fundamental process and the process science behind the manufacturing of lipid nanoparticles for mRNA delivery and the development and scale-up of these processes. You may recognize Don from a talk yesterday in the plenary session, and I'm very grateful here, Don, that you are here. It must be exciting times at Moderna on the COVID-19 vaccine. And I'm very pleased that you are here to show us some more insight on the mRNA. Actually, this was already scheduled before we had the COVID outbreak. So thanks, Don, the floor is yours.
Thanks, John. I appreciate the introduction and the opportunity to present. I do think this is a very exciting time for mRNA nanoformulation, so I hope I can provide some insight into that. Let's see if I can just get, okay. Let me start by providing a very brief background for folks who are unfamiliar with delivery of messenger RNA. Delivery of messenger RNA the vehicle has to serve a number of important biological functions to deliver its payload to the cellular cytosol. It needs to impart stability, and it needs to enable cellular uptake and endosomal escape. And these tasks, quite complex tasks, are typically performed using lipid nanoparticles or LNPs, although a number of groups have studied alternative approaches. I'm going to talk about LNPs today. LNPs are typically composed of several lipid components, including a cationic lipid, a zwitterionic phospholipid, a sterol, and some sort of PEG lipid conjugate. The cationic lipid is generally what's considered to be critical to LNP function, and the other lipids are believed to be present mainly to stabilize the particles. LNPs are commonly formulated using what is apparently a simple process in which an aqueous buffer that contains the mRNA, which has been pH adjusted to a pH such that the mRNA is a polyanion, is combined with an ethanol solution containing the lipids. And when that's done properly, the so-called solvent drop nanoprecipitation results in mRNA-loaded lipid particles with a size in the area of 100 nanometers. And when appropriately formulated, these particles can be reasonably effective for nucleic acid delivery in general, and many groups use them for mRNA delivery. As I said at the top, I think that nanoformulations for mRNA delivery have reached an important new stage in their development this year. For many years, investigators have worked to adapt the LNP technology, which was originally developed for delivery of smaller nucleic acid payloads like silencing RNA, and first commercialized for that purpose by Alnylam about 2 years ago to delivery of messenger RNA. There have been a number of challenges along the way, particularly in regard to creating a delivery vehicle with adequate potency and therapeutic index, and there are a number of pharmaceutical challenges that remain, for example, product stability. Use of messenger RNA to produce protein antigens for the purpose of immunization was the first widely studied clinical application of mRNA delivery, and 2020 has been obviously a year of rapid evolution in this area, with several firms bringing vaccines against SARS-CoV-2 to the clinic, and hopefully to licensure and distribution as part of the response to the pandemic. As part of the advancement of the mRNA vaccine platforms to licensure and commercialization, there are a number of activities which will need to take place, which have not been previously tackled for mRNA nanoformulations. Those include the detailed study of manufacturing processes required for successful licensure, the scale-up of those manufacturing processes to previously uncontemplated scales, and the refinement of analytical techniques to study in detail the differences between product manufactured across scale and site. The information from these studies will be shared with global regulators and with the scientific community, and out of this, I hope that we'll see some consensus emerge about key considerations associated with product quality for these types of formulations. And as we hopefully move past the pandemic, firms will begin to consider actions associated with improving the pharmaceutical acceptability of these formulations. And again, I would cite the example of product stability as a key concern. On the other hand, there's a growing body of non-clinical data with some emerging clinical data as well, which continues to highlight that mRNA therapies have an important opportunity beyond vaccines to improve human health. That said, clinical application of mRNA for therapeutic purposes is still in its infancy, and there are a number of challenges in this area related specifically to delivery, including development of suitable non-clinical models, demonstration of tolerability for frequently or even chronically administered therapies, and improvement of drug delivery to a broader array of target tissues and cell types. All of those have to be unlocked for therapeutic applications. I'm going to say a few more words about what I anticipate in the area of mRNA-based vaccines. Certainly, this is a pivotal moment for rigorous clinical evaluation of the idea that mRNA can serve as the basis for a safe and potent vaccine for a broad patient population. But if this hurdle is overcome, I anticipate that broad interest in the potential of this modality will drive important advances in scientific understanding. One area where I anticipate progress is around the process design, process control, and analytical evaluation of which is critical to licensure applications. I think we've already seen some important advancement toward a more fundamental understanding of critical quality attributes of mRNA vaccines, many of which have to do with the nanoparticulate nature of these formulations. I also think there will be significant learnings associated with scaling up these processes to truly industrial scales. Another topic which has been in the news recently is around the stability of mRNA nanoformulations and the need for cryogenic conditions to ensure product quality and the attendant complexities for distribution and use. While the fundamental thermal lability of messenger RNA is a liability which will remain, I anticipate that significant ongoing research to mitigate the impact and establish products which are stable for extended periods of time at temperatures that are more commonly available for a flexible supply chain. Similarly, optimization of some of the pharmaceutical aspects of mRNA nanoformulations, for example, to improve dosing convenience, will become increasingly important. And finally, as regulators receive submissions from multiple sponsors, a general appreciation of the aspects of pharmaceutical quality that will be developed and shared broadly. And all of these initiatives will help to improve the quality and utility of mRNA vaccines. I'd like to shift gears for a moment now and talk about the potential of mRNA nanoformulations beyond vaccines, which I think is quite compelling. Yesterday, Dr. Patrick Baumhoff provided a very nice overview of the potential of mRNA therapies in Stream 1 and showcased the potential of this modality to create antibody therapeutics. I put here an illustration which parallels an image that he used, which tries to show schematically that by harnessing the body's endogenous protein production machinery, a very wide variety of therapeutic biomolecules can be produced. These can include endogenous proteins, and I'll briefly discuss one interesting example, but can also include designed proteins with desirable properties. For example, it's possible to add to the sequence of a particular protein a transmembrane localization region, and in the process of transforming a molecule which in its endogenous state might be secreted to a membrane-bound state. This type of engineering can be quite difficult to achieve using other approaches. Another interesting example of the utility of mRNA therapies is enzyme replacement treatments. We're all familiar with the idea that enzyme replacement can lead to significantly improved outcomes for patients, for example, with genetic enzyme deficiency conditions. However, it is generally not considered practical to design replacement therapies for intracellular enzymes. But the panel in the middle illustrates how mRNA can overcome this issue. This is non-clinical data generated by our clinical candidate for methylmalonic acidemia. Methylmalonic acidemia is a rare and serious metabolic disorder in which the methylmalonyl coenzyme mutase enzyme is either absent or not fully functional. This enzyme is an important component of the metabolic pathway for protein and fat metabolism, which takes place in the mitochondria. Because of the inaccessibility of this organelle to systemically administered protein, this deficiency was previously considered undruggable. However, an mRNA therapy can produce the endogenous protein within the cell, including the mitochondrial localization sequence, resulting in appropriate correction of the metabolic pathway. In the center panel, you'll observe a dose-dependent restoration of biological function, here represented by the reduction of circulating methylmalonic acid, which is the species that accumulates systemically as a consequence of metabolic failure. The mRNA therapy can reduce levels of the biomarker effectively to zero and can sustain this reduction following repeated dosing, even in conditions where complete lack of endogenous enzyme function. In the right panel, you can see the dramatic therapeutic benefit which can result in which the fatal phenotype of the enzyme deficiency, shown in the light blue trace from the Kaplan-Meier plot, is fully rescued following mRNA administration. The capability of mRNA therapies to achieve these kinds of dramatic therapeutic gains for conditions which are quite difficult to achieve using other therapeutic modalities has been demonstrated with increasing frequency in the non-clinical setting, making this a compelling area for further research. However, only the first steps for clinical proof of concept have been obtained, and therefore the potential for mRNA therapies in the clinical setting has considerable distance to travel before it can be considered demonstrated. For mRNA nanoformulations to succeed in the therapeutic arena, a number of challenges must be overcome, which are generally considered to be higher hurdles than those faced by vaccines. Many of these challenges are related to the formulation itself and may therefore be addressed or mitigated by the modification of the nanocarrier. One example is that in many cases, significant levels of therapeutic protein must be produced in order to generate a therapeutic effect, consequently requiring administration of higher levels of the mRNA therapy than required in a vaccine setting. The potential for these formulations to be immunostimulatory is a well-appreciated concern. Unless the formulation is carefully designed, this immune response can lead to the formation of anti-drug antibodies, in turn leading to rapid clearance and loss of product potency after multiple doses. Biodistribution of the protein produced and the mRNA administered may also be critical, both at a tissue level and at a cellular level, and the potential for the nanoformulation to modulate this biodistribution is an active area of research for many teams. Additionally, in contrast to vaccines, where a single administration may be sufficient to achieve sustained immunological protection, it will often be the case that a therapeutic mRNA will need to be administered repeatedly and potentially chronically. Naturally, the threshold for appropriate safety and tolerability of the therapy is quite high. I'd conclude by stating the obvious, that the requirements for a consistent, highly controlled manufacturing process are therefore strenuous. I hope I've helped to illustrate some of the complexities of what I think is a very exciting time for mRNA nanoformulations. The potential of these formulations as a weapon in the fight against SARS-CoV-2 has put them in the public spotlight and has created a whole new set of challenges for this modality as it progresses to licensure and broad use. At the same time, mRNA has the potential to serve as an effective therapy for many conditions beyond vaccines, but we're still on the threshold of demonstrating such potential clinically. The time ahead is very exciting. As the American baseball player Yogi Berra said, when you come to a fork in the road, take it. Thanks for your very kind attention.
Wonderful. Thank you very much, Don, for an excellent overview of the progress that Moderna has already made in the past to bring these mRNA products to patients, and to see the progress and the speed that you currently have in developing the COVID-19 vaccine. Excellent story that really takes us, let's say, to a next level, being put into patients. And I think that's also the link to the next speaker. Thanks, Don. Any question for Don, please, in the window below, and we'll come back to that at the end of the session. Thanks again, Don. Our next speaker is Dr. Beat Vlieger. Beat is a pharmacist by training, and he has a PhD in molecular biology. Beat has ample of experiences in different pharmaceutical industry, and he is at present a global lead nanomedicines at Vlieger Pharma, based in Switzerland. Beat is a member of the NBCD working group, and I'm sure that after all the sessions we just had on manufacturing, producing, technology, Beat's going to focus on the practical implications and how to actually use these type of products. Beat, please, the floor is yours. Maybe you're on mute.
So, thanks, Jan, for the introduction.
Wonderful.
As Jan mentioned, I have the pleasure to tell you something out of our experience with having this type of nanomedicines in the market for about 50 years and more. So I'm really have the topic here from manufacturing to bedside. What can go wrong, or what do we have to have a special attention to looking at that? So actually looking into that, I was following the FDA's draft guidelines for nanomaterials and looking at all the problems that we are facing there. It starts actually with the adequacy of characterization of the material structure and its function. We heard a lot about that. We also need then to understand the mechanisms by which the physiochemical properties of material impact its biological effects. I think this is a part where we have lots of question marks still with these products. Then further, we need to look into physical and chemical stability. For that, we need to understand point 1 and 3. And as a last point, physical state of the material upon administration. Again, here, products can look completely different after injection or even after handling. And for that, we were actually looking in what's happening and what type of literature is available for our type of products, which are the intravenous iron products from manufacturing to the bedside. So we started with the production. And here, actually, that's something we need to have under control, from the source of materials to production procedure, conditions of autoclaving these complex materials. Primary packaging can even influence, as we learned, batch-to-batch reproducibility, a key aspect, and scale up. If we go again back to this draft guidance of the FDA, this is also mentioned here. These products are very sensitive to process conditions and production scales and need a lot of attention if we have to do a manufacturing change. And if you have a product launched and you start to sell it, after 10 years, you have to change your manufacturing process. So there are several manufacturing changes which you have to control. Why are they so critical? Because the surface of these particles are complex and they define actually the biodisposition, the pharmacokinetics, and the pharmacodynamics of those products, which are essential in the clinics. Now, if we have a very simple molecule, we just look at the composition, at the chemistry. Here, we have complex molecule consisting of an iron core and surrounded by carbohydrates. These carbohydrates are already complex structures, and we need to analyze the whole structure of this product, the whole particle, with all his surface functionalities, with the size, with the shape, and so on and so forth. So these are the critical parameters we need to check during manufacturing. We need to make sure whatever we change in our manufacturing process, none of these aspects is affected. Now, if you go on, transport, temperature, moisture, light, time, and even vibration of these particles can have an effect on their stability. So storage and transport are key elements which we need to handle. And remember, we need to have a storage of up to 4 or 5 years. People have them, or clinics have them, sometimes not under the most ideal conditions. Transport, we might face very harsh conditions for these products. What is very special about these particles is actually that it's not only the chemical composition which is relevant, but what we see in aging or in storage, we can either have an aggregation of the particles, which is not desired because it also changes the kinetics and the stability, or we can have a decomposition of the particles, which for sure is not the idea of these particles. As you can see, this is not theory. These are actual samples we found in the market, which were not properly handled. And then you end up with an injectable, which you for sure not want to have injected into you, because these particles were physically falling apart. Now, the product handling is another point which is actually very astonishing and has some consequences. In a hospital pharmacy, in many places, actually infusions were prepared from these stock solutions. So they are made in the hospital pharmacy, then shipped out to the wards, to the individual wards, and the patient might receive it 4 hours later, 5 hours later. That's not so clear in any case. So we need to make sure that even if we dilute these particles in a diluent, that they are still stable, and we can guarantee that the patient gets the right drug injected or infused. So there are many aspects here we need to consider. And again, we have here examples of 2 products which are actually from the same iron sucrose. Both are complying to the USB specifications, but they are from different manufacturers. So what we see here already, they have different colors. So something is not the same. If you look then at the stability of those in terms of particle size, you see also that after dilution, those particles are changing size on some of these products. Does this matter? That's actually the question now. Is this just something we measure here? Is this really a critical quality attribute? Or are we having substantial changes in the clinic?
2 minutes, Beat.
Yes, good. What we can see in one of the studies in the clinics, in a retrospective study, they injected gynecological patients with these drugs. And what they did, they had some side effects and wanted to get rid of the side effect and further diluted these products, which were further destabilizing the products. And what you can immediately see, there was an increase in side effects. Now, as I said, the manufacturing process defines the product, but that's not everything which is relevant, but also the physicochemical characteristics can be affected by the stability during storage or preparation of the ready-to-use products. This can have an effect on several parameters like particle size, surface properties, etc., which impact stability, the pK, adverse events, and immunogenicity in worst case. So it has an effect on quality, efficacy, and safety. So in summary, we were looking at all the data which were published in these areas, and we actually could find that there is not a lot of data out there, and there is a huge gap of data in terms of storage, in terms of how to dilute, with what to dilute your products, although we know that it is absolutely critical for this type of products. With that, I would like to thank you for your attention.
Wonderful. Thank you, Beat, for this presentation and for staying in time as well, as is always important in CLINAM sessions. I think you nicely showed that we can design all fancy and complicated type of products in the end. If we follow all the regulations and if we go through the full process, we leave it to the hands of the end user, the hospital pharmacist, the healthcare professionals, or other doctors. And if they have a certain awareness of the challenges with these type of products, I think it will really be to the benefit of our patients. So questions to Beat, please list them below and we'll ask them after the next speaker. So I'm very pleased to announce our next speaker in this session. It's Dr. Wenlei Jiang. She serves as a senior science advisor in the Office of Research and Standards, in the Office of Generic Drugs of the Center of Drug Evaluation and Research from the Food and Drug Administration in the US. I think we've nicely seen the different products coming through. And in the end, of course, as a system in general, we're looking towards generic products, biosimilar products, etc., to also play into the cost effectiveness of different pharmaceutical products. Wenlei has a leading role within FDA and also in the PQRI and in the global harmonization discussions on how to deal with generic complex drug products. So I'm very pleased that in the end of the session, we'll come to this phase. In the end, it's inevitable that some of these products become generic, and that's a good reason. But how do we do that? Wenlei, please, the floor is yours.
Yeah, thank you, Jan, for the kind introduction. Today, I'm going to talk about complex drug products containing nanomaterials. So the views expressed in this presentation are mine and should not be interpreted as FDA policy. I think complex drug product is not new to everyone. The complex drug product concept I'm going to discuss today was first introduced in GDUFA 2 commitment letter. GDUFA stands for Generic Drug User Fee Amendment. Generic drugs are copies of brand name drugs. GDUFA provides user fees for FDA to ensure timely review of applications for generic drugs. So in the GDUFA 2 commitment letter, FDA provided a list of complex drug products, including products containing complex API, complex formulation and dosage form, complex route of delivery, complex drug device combination. Here, I want to emphasize, in the context of GDUFA 2, complex products are the ones which present challenges for generic drug development. That is difficult to assess drug substance sameness and or bioequivalence at the site of action. In the past several years, FDA have been developing complex product classification criteria so we can standardize the complex product classification process, understand the complex product landscape, and prioritize regulatory research to support complex generic drug development. This slide provides more details on how we classify certain products as complex product. For example, any drug product containing a complex API, regardless of its administration routes and dosage forms, is considered a complex drug product. Any non-oral formulation dosage form, where there are often 2 or more discrete states of matter within the formulation, is considered complex dosage form and formulation. Here, non-oral nanotechnology products fall into the category of complex dosage form and formulation. They include like nano-sized liposome, nano suspension, lipid complex products, and others. Here, I do want to clarify that oral tablets containing drug nanocrystals are not considered as complex product within GDUFA-2 because a conventional in vivo PK study can be used to demonstrate bioequivalence for these products. In the past 20 years, FDA has approved a number of parental nanotechnology products. However, we have limited generics available for these products. Currently, we have 3 generics approved for doxorubicin hydrochloride liposome injection and 1 generic approved for sodium ferric gluconate injection. There have been significant challenges in developing the generic version of these parental nanotechnology products because of their size and higher order arrangement. Recognizing the complexity of nanotechnology products, FDA published a series of guidances starting from the basic one, providing clarification about which product are considered the ones involving the application of nanotechnology, and an overarching guidance covering drug products, including biologic products that contains nanomaterials. Besides this general guidance, FDA also published guidance related to liposomal drug products because liposome accounts for the most common form of nanotechnology products. In addition, to guide generic drug development, Office of Generic Drugs publishes product-specific guidance. The guidance at the bottom serves as the foundation of regulatory framework. The ones at the higher level have increased specificity for products. Here is an example of product-specific guidance for doxorubicin hydrochloride liposome. We recommend the generic formulation to be qualitatively and quantitatively the same as a brand product, using the same remote loading manufacturing process, equivalent physical chemical characteristics as the reference drug, and also equivalent pharmacokinetics of unencapsulated and liposomal associated drug in vivo. Here are more product-specific guidance we developed for other nanotechnology products. Besides guidance development, FDA also has an active nanotechnology regulatory research program focusing on quality, safety, equivalence, and post-marketing surveillance of nanotechnology products. This research program is to foster the nanomaterial development and establish the tools, methods, and the data to assist in regulatory decision-making, while also provide in-house scientific expertise and capacity. I will highlight some of our research efforts in the development of in vitro characterization methods for nanotechnology products. Here, we developed a capillary electrophoresis method, which provides simultaneous separation and quantification of unencapsulated doxorubicin and liposomal encapsulated doxorubicin. This method overcomes the issue of drug leakage and the lengthy separation process encountered in the conventional solid phase extraction method. The second example is AF4 method to separate liposome particles into different sizes and simultaneously quantify the drug and excipient content using HPLC-MS in liposomes with different size. This method provides a more comprehensive and in-depth characterization of liposomes compared to the conventional analytical method. If you want to know more about CEDAR research related to nanotechnology products, please refer to these links. In recent years, we continue our efforts in promoting harmonization of evaluation criteria for nanotechnology products through engagement with IPRP Nanomedicine Working Group and the Global Bioequivalence Harmonization Initiative conferences. Regarding product-specific guideline of doxorubicin hydrochloride liposome injection, previously EMA and FDA differ significantly in terms of moieties to be measured in bioequivalence study. Now we have achieved a high degree of agreement. We decide to measure the unencapsulated and liposome encapsulated doxorubicin for bioequivalence demonstration.
2 minutes, Wenlei.
Yeah, last one. So in summary, non-oral nanomaterials are considered complex drug products, and there are limited number of generics available for these products. So far, we have doxorubicin hydrochloride liposome injection and the sodium ferrochlorugluconate injection generic versions available. US FDA publishes guidances and conducts regulatory research to support nanotechnology drug product development. International collaborations are needed to facilitate research and standards development, as well as harmonization of regulatory recommendations for nanotechnology products. Finally, I would like to acknowledge my colleagues from Office of Research and Standards, Office of Generic Drugs, Office of Pharmaceutical Quality, and Office of Regulatory Affairs who have contributed to the work I presented here. Thank you for your attention.
Thank you very much, Wenlei, for this overview of all the activities within FDA, intramural, extramural, and on an international level to, let's say, facilitate the approval of generic products of these complex drug products. Very impressed always, especially by the categorization of the complexity of the products, because you manage to kind of chop up the larger issue into smaller parts that indeed make sense. So thanks so much. I think we are here at the point where I switch to my co-chair, Scott McNeil, to address the speakers and the audience with the questions. Scott, please.
Okay, thanks, Jan, and good evening from Basel, or wherever you're at. Hopefully it's not too late or too early there. We still have about 15 minutes for questions and answers. So as Jan said, we encourage you to submit your questions in the Ask Question button at the bottom of the screen. So we heard from, in this session, we heard from investigators ranging from academia clear through full process development and the pharmaceutical industry. So we've got quite a selection that we can ping and ask questions from. So let me start with one while everyone else is submitting their questions. We heard terms such as CQAs today, and the process is the product. So one could imagine in trying to define the CQAs for a specific product, you could generate quite a huge data set due to physical chemical characterization and biological data and so forth. So coming back to something that Don said in his original presentation, he said the technology was already there in 2000. But my question to the group, especially in light of this topic, is have the CQAs changed, especially with some of the follow-ons, due to the technology, due to the knowledge that we've gained over the last 20 years? So I'll submit that to the panel and await your responses.
Do you have a preference for someone to answer this one, Scott?
You know, I'm waiting for them to be brave and raise their hand, and then I'll call them Don.
Dan is waving at us. Please unmute yourself, Dan, and address the question.
Yes. Okay, that's a good point. Of course, I try to be a little bit provocative, and apparently it worked. I remember that in 2002, we had a meeting with members from the European regulators and EMA and FDA to come up with guidances on liposomes. And in that meeting, a number of issues were discussed. And on that basis, and I guess on the basis of their own experience, the FDA drafted, I think in 2002, something like 2002, a draft guidance that has been in place. Wenlei, help me if I'm wrong and make a mistake there, has been in place, I guess, until 2018, something like that. And when you compare the text of the draft guidance from the early 2000s and the final guidance that was released in 2018, something around the time, then yes, there are more details there, and it is worked out in more detail. But basically, the draft guidance and the guidance in 2018, they are not that different. So over the years, the characteristics of the liposomes that were sort of requested to come up with, they haven't changed much. What I noticed, however, is that the terminology and that is used, and in particular the critical quality attributes and the design space and the literature on that has sort of exploded. And I sort of consider that as something that had to happen, because in the end, you can put all these guidance and documents there, and you can list a long list of parameters that you have to measure, and FDA has done that in quite a number of times. And my point was always, well, which ones are important and which ones are not? Well, and those simple questions are now translated in critical quality attributes. And I think that's progress. That is, we have this long list of things that we can measure, but that's not important. It's the ones that really count. And in that sense, over time, things have changed. The long list was there already a long time ago, and now we focus on the things that are really important. So, Wenlei, I think it's now up to you to sort of comment, shoot me down.
I think, Jan, you have asked this question at a number of meetings. I think this is a very good question. I thought I have addressed this question before, but yeah, I don't mind saying it again. I think for FDA, we publish product-specific guidances for different drug products to guide generic nanotechnology product development. In those guidances, actually, we listed specific quality attributes. We ask for comparative evaluation, such as particle size, the morphology, the in vitro drug release. So we do consider these as critical quality attributes for comparison. Yeah, so I feel like in our PSG, we have listed the critical quality attributes for both the generic and the brand manufacturers to pay attention to measure those attributes carefully and do the comparative evaluation.
Okay, good, thanks. Now maybe a question for Don and Mark. As you're scaling up, as you're going to clinical trials, one is almost fixed into the partner that you have in your phase 3 production lot. So my question relates to Mark. Do you have the capacity for very large scale doses? And to Don, how are things like IP handled for that type of scale up? How do you partner? Is it an issue of trust, or do you leave it to the lawyers for that to happen?
I can take the first part of the question, so thank you. So you're right that, of course, you don't want to make changes during the various clinical trials that you're doing. But I do think the technologies that we employ are amenable to making larger batches also. So of course you need to gear to what you need at the various phases. But the technology would be the same for making larger scale batches. And the question whether we have done this, so we have made batches, I think, of up to 300 liters. Depending on the kind of market, very small markets, I think 300 liters could be commercial. Larger markets, you might be looking at different manufacturers. Ardina is really focused on early clinical development, so taking a preclinical program and bringing it to the clinics. So that's what we are, I think, most experienced in.
So Scott, I'll just briefly address the second part of the question. I think leaving it to the lawyers is not a very successful strategy. Ultimately, to have a good relationship with a vital partner, you have to be transparent, and you do need a degree of trust. I think understanding the process for manufacturer and the analytics associated with it is vital for a CMO to do their job properly. So we do extend that trust in our relationships.
Okay, great. Thanks, guys. So coming back to the CQAs, a related question that we get often is that nanotech is just simply a new branding. So my question to many of the producers and those of us in between is how important is the nano aspect to your API? More specifically, is it germane to the formulation and contribute to the CQAs, or is it just an excipient? How are you handling that, and how are your partners viewing that, and even the regulatory agencies? So how important is that nano carrier to the ultimate formulation, whether that just be due to size or the packaging, encapsulation, and so forth. Beat, we haven't heard from you. Any comments on that?
Yes, I thought that's probably my topic. There are two aspects. The whole term nano is in some countries has also a very negative connotation. So in the past, it was actually avoided to use the word nano, and at least in German-speaking areas. Then the importance of nano, I think this is one of the very fundamental things of at least our iron carbohydrates. This nanostructure makes it possible that they are active, that they go to the right part, and that they are not toxic first and foremost. And I think these nano characteristics, far beyond just size, but also the surface structure of these particles, is absolutely key to their kinetic and actually toxicity profile. So for us, this is one of the key elements that they are nanoparticles. It goes, I mean, iron is iron. That's a simple molecule. But to deliver it to the body in a way the body can use it and it goes to the right place, that's the trick of the nanoparticle here. So I think it's not just a formulation, it's actually far more than a formulation. It's the drug.
Okay. Suzanne, just a quick question for you as far as your databases and your algorithms. What I've noticed in my case, the literature usually lacks or is, I'm sorry, the other way around. The literature is usually in front of what we're seeing in approvals and commercialization. And sometimes it almost takes that approval and commercialization for information to become public. So how do you account for that in your searches and just the knowledge that JRC and the EC are gaining as you're tracking this field?
I'm not really getting your question.
Yeah, so when you're doing literature searches, sometimes the literature is 5 to 10, maybe even 15 years ahead of what is being commercialized.
Yeah.
And so is there a process where you can inform that even the approval of some of those incoming products?
I'm sorry, Scott, I don't know. Do you see me? Do you hear me?
Yes, we do. Okay, it's all right, Suzanne. There is a question that came in from, let me go ahead and move on to one of the online questions. It's from Bernd. So he's saying that the best-selling nanomedicine seems to be paliperidone, palmitate, nanocrystals, and then LAI, long-acting injectables. It's about a $3 billion market. So is there a general benefit for nanosizing LAIs or something like microsizing LAIs? And I think that is probably very influential as far as nanocrystals, but any other thoughts on the advantage of a nanosize versus a microsize? Because I can answer it. So there's certainly aspects with particle size, as you've heard today and yesterday, as far as different biodistribution and so forth. Bernd, I hope that we can, in the various sessions, we've answered that question. And it's certainly far more than just size now, that there are aspects of surface chemistry, release kinetics, and so forth. It's as you point out, the nanocrystals are almost a different field than some of the third and fourth and even fifth generation particles. So I can see that our time has expired. Any other following remarks before I hand it over to Jan? Okay, Jan, I'll turn it back over to you.
Thank you, Scott. I've seen one more question. Let's try to ask that as well, and which was to Mark. Mark, any process analytical technology that you have standardly incorporated in your manufacturing process?
So the question is, if we have standard methods that we apply, I think methods like residual solvent are very general. So we have standardly validated methods to do this. Of course, we need to confirm suitability for each product because any product has a different matrix, so it could affect the validity of the analytical method. But to my understanding, that kind of methods have always worked. Other methods would be, I think, the pharmacopoeia methods, of course, that are standardly available. And of course, there is parallels in lipids for liposomes if you have, but it's always about the composition and being able to separate all the various components. So in some cases, you might have a good starting point, but you still need to check for a new combination of lipids that it's an acceptable separation.
All right. Thank you so much. Wenlei, you wanted to comment?
Yeah, sure. Actually, Mark, that question is from me.
Thank you.
Clarify whether you have implemented any in-process analytical technology in your manufacturing process. Not only
final release, you mean? So besides final, yeah, absolutely. This is the way how we control our manufacturing strategy. So we check size during the various steps. So immediately after preparation, after purification. So dynamic light scattering is, of course, a very easy in-process tool also. but also composition. So yes, we use in-process methods during the manufacturing process.
So you have those tests online or offline?
Okay. I think pretty much all of them are offline. Some are rapid, and that's why they are nice for in-process. But I'm not aware of some technologies that can measure, for instance, particle size really in your reactor. But so far, we've not really seen the huge benefit of doing this. So normally we check after, so we standardize the process parameters and in that way control the process. Yeah.
Okay.
Thank you.
Wonderful. If there's any further discussion, I ask you to take it offline if it is possible at all in this online world. I think for the sake of time, I think we have to close the session. There's a plenary session starting in a minute from now. I'd really like to thank all the speakers, all the attendees, CLINAM organization for hosting this science-based discussion. And I'm really looking forward to seeing you all face-to-face next year, hopefully, and otherwise, let's meet each other online. Thanks so much and talk to you soon.